तो रेस्पिरेशन चैप्टर हम करते हैं और एक रेस्पिरेशन का, तो जब रेस्पिरेशन चैप्टर हम करते हैं बोले यार, तो उस रेस्पिरेशन प्रोसेस में हम क्या देखते हैं कि हमने ग्लूकोज कंज्यूम किया, हमने खाना खाया तो ग्लूकोज लिया, उस खाने के साथ हमने ऑक्सीजन कंज्यूम किया रेस्पिरेटरी सिस्टम के थ्रू। जब ये दोनों आपस में मिलकर काम करने लगे तो उससे हमें क्या मिला, CO2 एक्सहेल कर दिया हमने, H2O बॉडी में यूटिलाइज हो गया, और साथ में ATP या फिर एनर्जी मिल गई, और उसी एनर्जी को हम यूज करते हैं आज, है ना। कुछ भी हम काम कर रहे हैं तो एनर्जी चाहिए, और एनर्जी खाने के टूटने से ही आती है, फूड के ब्रेकडाउन से ही आती है। ठीक है। अब हमें बैलेंस भी करना है कि अगर लेफ्ट साइड में 6 कार्बन है तो राइट साइड में भी 6 कार्बन होने चाहिए, है ना। लेफ्ट हैंड साइड में 12 हाइड्रोजन है तो यहाँ पे भी 12 हाइड्रोजन होने चाहिए, तो बैलेंस हमने कर दिया और एनर्जी प्रोड्यूस हो गई। अब रेस्पिरेशन की प्रोसेस का मतलब क्या होता है कि एनीबडी हु, जो सारे हिंदी वाले लोग हैं ना, ठीक है। तो अगर हम इस प्रोसेस के अंदर देखें तो होगा क्या कि ग्लूकोज जब ऑक्सीजन के साथ रिएक्ट कर रहा था तो हमें CO2 H2 और एनर्जी मिल रही थी, और इसी एनर्जी को हम यूज करके काम करते हैं। अब जो हम 10th क्लास तक पढ़े हैं उसमें हमने ज्यादा फोकस यही किया कि हम सांस लेते हैं, हमें ऑक्सीजन मिलती है और हमने एनर्जी प्रोड्यूस कर दी। यहाँ पे वो पूरी प्रोसेस होती क्या है, इसी रेस्पिरेशन की प्रोसेस को हमने मल्टीपल स्टेप में डिवाइड किया है। पहला स्टेप है ग्लाइकोलिसिस, कि हमने ग्लूकोज जब हमारी सेल के अंदर एंटर किया तो उस ग्लूकोज का ब्रेक डाउन हुआ, और जो ग्लूकोज है वो किस में कन्वर्ट हो गया, पायरुविक एसिड में।
तो सॉरी टू इंटरप्ट यू, हम रिकॉर्ड कर रहे हैं ना?
हाँ,
बट फिर मतलब अगर हिंदी में करेंगे तो फिर बाकियों को समझ नहीं आएगा।
अगर जो क्या बोलते हैं,
better to go with English, Hindi plus English.
ओके, मेक सेंस। सो फर्स्ट स्टेप इज ग्लाइकोलिसिस इन केस ऑफ रेस्पिरेशन प्रोसेस, जहां पे ग्लूकोज पायरुविक एसिड में कन्वर्ट होगा, एंड वी हैव 10 स्टेप फॉर दिस। सेकंड, लिंक रिएक्शन। In case of link reaction, this pyruvic acid will convert into the smaller unit acetyl coenzyme A. So here 6 carbon convert into 3 carbon. Here 3 carbon convert into 2 carbon. Third step is Krebs cycle. This Krebs cycle is also known as TCA cycle, tricarboxylic acid cycle, or citric acid cycle. Citric acid cycle. In this case, what happen when this acetyl coenzyme A react, it will produce CO2, but along with CO2, example, one acetyl coenzyme A have 2 carbon, so 2 will have 4, here you will produce 4 CO2, correct? Here 2 pyruvic acid, 2 acetyl coenzyme A plus 2 CO2. हेलो, भूल गया था मैं। तुम हैं? एक मिनट, पढ़ा तो मैं गिर दिया हूँ। हाँ, तो भाई
तो वो क्रेब्स
ग्लाइकोलिसिस,
साइकिल के बारे में क्या बोले प्रोसेस थोड़ा बताएंगे?
in Krebs cycle, see, one glucose convert into 2 pyruvic acid, correct? Because glucose have 6 carbon, so one pyruvic acid should have 3 carbon. So 3 carbon to the 6 carbon balance. These two pyruvic acid convert into acetyl coenzyme A and CO2. Acetyl coenzyme A CO2 में convert होंगे, साथ में यहाँ पे NADH बनेगा, साथ में FADH बनेगा, and we will produce ATP. Correct? And last process is electron transport system. Electron transport system, if electron pass from one system to another system, ATP will produce. Example, NADH. NADH will produce 3 ATP, 1 NADH. 1 FADH produce total 2 ATP. Correct? So ATP should not produce ATP. That's why you will see in this process total 3 to the 6. So total 6 NADPH is produced here, 2 FADH. Correct? So if 1 NADH produce this, in this process here you will have 2 NADH. In Krebs cycle there will be 2 NADH. What is the total number of NADH in the whole process? 2 in glycolysis, 2 in link reaction, 6 in Krebs cycle. Total NADH, total number of NADH consumed in the whole process is 10. Total number of ATP it should produce 3 into 10, 30. Total number of FADH, if you see, 2 FADH is produced. Number of ATP from this 2 to the 4. And 4 ATP direct. 4 ATP direct. Total number of ATP produced in the whole process is 38. 38. Done. So this is the whole chapter we need to read about. हमें यही पढ़ना है कि पहला स्टेप होता है ग्लाइकोलिसिस. हाउ इन केस ऑफ ग्लाइकोलिसिस pyruvic acid produce and 2 NADH produce. In link reaction, how acetyl coenzyme A and 2 NADH produce. Krebs
So
cycle.
I'm joining within 10 minutes.
Okay. And if you talk about ATP as well, 2 ATP are produced here. 2 ATP will be produced here. So total 4 ATP. Correct? That's what direct ATP. Total number of ATP 30. If anybody asks why are we reading this chapter, क्यों पढ़ रहे हैं हम ये सब, उसका रीजन ये है कि जब हमें समझ में आया when we got to know that कि mitochondria produce energy, then we can manipulate it. The Red Bull, Sting, tea, coffee we are consuming is the actually manipulation of the same system. Make sense? Any doubt? This is the summary of whole chapter. Namita, this is what you were looking for.
हाँ सर.
So when we got summary, now I can explain each topic one by one. First step is glycolysis. In case of glycolysis, what will happen? We consume glucose with the help like food enter in our body. खाना खाएंगे और ग्लूकोज हमें मिल जाएगा. Correct? तो in case of glycolysis, we know it is glucose. Lysis means breakdown. Glucose breakdown. This word come from the Greek. This glycolysis pathway given by 3 scientists, Amden, Meyerhof, and Parnas. Amden, Meyerhof, and Parnas, 3 scientists gave this pathway. That's why glycolysis, another name is EMP pathway. And glycolysis occur in cytoplasm of the cell. Cytoplasm of the cell. And it occur in all living organisms. It occur in all living organisms. Every living organism have this process, whether aerobic process or anaerobic process. अगर ऑक्सीजन है तब भी ये काम करेगा, ऑक्सीजन नहीं है तब भी काम करेगा. अगर एनारोबिक प्रोसेस है, इन केस ऑफ एनारोबिक प्रोसेस, अगर ऑक्सीजन नहीं है तो ओनली मेथड ऑफ रेस्पिरेशन. दैट्स व्हाई यू विल सी इफ वी आर ह्यूमन बीइंग, हम इंसानों को हम सारा दिन खाना खाते रहते हैं, है ना. तो वी हैव अ हैबिट ऑफ ईटिंग टू मच, क्रेविंग, नाइट क्रेविंग वी वर टॉकिंग अबाउट. तो अगर हम सारा दिन खाना खाते रहते हैं, उसका पर्पज क्या है? क्योंकि हमें बहुत एनर्जी चाहिए, वी नीड लॉट ऑफ एनर्जी. हाउ डू वी गेट दिस एनर्जी इज एक्सप्लेन बाय दिस चैप्टर. यही चैप्टर बताता है जब हमने खाना खाया, उस खाने से एनर्जी कैसे मिली, है ना. और उस एनर्जी यूज कैसे किया वो सेकंड चैप्टर, बट एटलीस्ट एनर्जी कैसे मिली. हम वॉक कर रहे हैं, रन कर रहे हैं, सो रहे हैं, सबके अंदर वी नीड एनर्जी एंड दैट एनर्जी इज प्रोवाइडेड बाय दिस रेस्पिरेशन प्रोसेस. Understood. Now, the process is, what is the main conclusion of this topic? Glycolysis occur in cytoplasm when you have glucose inside the body. This glucose will break down into the smaller molecule, that is pyruvic acid, 3 carbon. In this topic, we also produce 2 NADH, 2 net ATP, and 2 H2O molecule. Any doubt? Question is how to produce glucose, how we get glucose. We get glucose from the starch, but in case of plant, because this chapter is about the plant, plants store their food in the form of sucrose. Correct? Plant store their food in the form of sucrose. Sucrose will convert into glucose plus galactose. Correct? So now we will study how this glucose will break down into the different parts. Example, first you have glucose. So glucose will convert into glucose 6-phosphate. Yes.
ग्लूकोज प्लस फ्रुक्टोज होगा ना, नॉट गैलेक्टोज.
Yes. My bad. मैंने क्यों लिखा है ये? I don't know. Glucose plus fructose, and enzyme used in this case is invertase enzyme, because invert sugar. Sucrose is invert sugar, that's why invertase enzyme. So first step is glucose will convert into glucose 6-phosphate. हमने phosphate add किया, we added phosphate. So some enzyme is involved in this case. Enzyme name is hexokinase. Why? Because this enzyme act on 6-carbon glucose. That's why hexokinase. First step done. Glucose 6-phosphate convert into fructose 6-phosphate. As you can see, glucose and fructose have same formula. So here you have isomer. That's why isomerase enzyme will act in this. Because both have same carbon. Same carbon compound are isomer, and isomer enzyme isomerase. Now you want to add one more
सर
phosphate in this. Yes, Anvita. Pratigya.
मेरा कुछ ज्यादा ही नेटवर्क इश्यू हो रहा है, यहाँ पर हवा चल रही थी, तो आप मुझे क्लास के बाद नोट सेंड कर देंगे प्लीज.
ठीक है.
स्टार्टिंग से अच्छे से कुछ नहीं लिखा.
ठीक है. तो यहाँ पे what will happen guys कि because we are adding phosphate, how we can add phosphate? Because introduction of ATP. जब ATP ऐड करेंगे तो वहां से phosphate निकलेगा. That phosphate will become 6-phosphate. Fructose 6-phosphate will convert into fructose 1,6-phosphate bisphosphate. Fructose 1,6 bisphosphate means 2-phosphate. Again, how we got second phosphate? Because of ATP. Wherever you have ATP, this step is important. Correct? This fructose, if you say, it will convert into 2 parts: dihydroxy acetone phosphate. Dihydroxy acetone phosphate. It is known as DHAP, correct? And there will be phosphoglyceraldehyde. Phosphoglyceraldehyde. Al means aldehyde. So it means, and both of these can convert into each other. Both of this can convert into each other. This into this, this into this. So imagine कि हमारे पास दो मॉलिक्यूल PgL के ही है. यहाँ तो कोई डाउट? पहले we added phosphate with the help of ATP. Isomerization, glucose into fructose. Then again ATP added, so 2-phosphate, and it convert into the PgL form, phosphoglyceraldehyde. Next step is really important. Why? Because now this is aldehyde, and when any aldehyde convert into, like, undergo oxidation, then you know it will convert into what? Carboxylic acid. Carboxylic acid. Same thing here. When you introduce NAD positive chemical, in this case, it will convert into NADH and H positive. Very important. What is special in this case? Oxidation का मतलब क्या होता है? Addition of oxygen or removal of hydrogen. In this case, we are removing hydrogen. That's why this is oxidation. This process is oxidation. In this oxidation process, and it will be bis phosphoglyceric acid. Bis phosphoglyceric acid. Again 3 carbon, but very important molecule NADH is produced in this case.
So I'm so sorry for joining late. I wasn't feeling well.
It's okay. Now you're feeling well?
Yes, sir.
Okay, good. So now, as of now, whenever we were adding phosphate, ATP was consumed. Now we will remove phosphate. What will happen? ATP will be produced. So BPGA, when convert into 3-PGA, it means we removed one phosphate. So ATP will be produced. And because you take 2 molecule every time, so what will happen? 2 molecule of this, then 2 NADH will be produced, 2 ATP will be produced, because one step, 1 ATP, 2 step, 2 ATP, 2 molecule, correct? Then isomerization, third position of phosphate convert into the second position. Not really crucial, correct? Then this second position will convert into phosphoenol pyruvate, same word you have heard in case of photosynthesis chapter. Phosphoenol, in and all, alkene and alcohol. So it happened because of the removal of water. So 2 molecule of H2O will be removed in this case. And last step, where phosphoenol pyruvate convert into pyruvic acid. 3 carbon molecule, 2 molecule, and because you removed phosphate part, again 2 ATP will be produced. Any doubt, guys? And this is the summary of the glycolysis. Ultimately, what is the purpose? Breakdown of big molecule glucose into the smaller molecule pyruvic acid. Breakdown. Ultimately, breakdown from this to this. In this process, 4 ATP will be produced. 4 ATP will be produced, 2 NADH will be produced, and 2 ATP consumed. Any doubt, guys? So we already said this. When this process of glycolysis happen, what will happen? 2 molecule of pyruvic acid 2 molecule of NADH, 2 ATP net, 2 ATP net, and 2 H2O molecule. Any doubt, guys? Was it really difficult? If you read this once or twice.
यूपीएस से difficult है.
Any doubt?
No, sir.
So, why we are reading about this chapter? How glucose breaks down into the smaller molecule, because we will further discuss this smaller molecule. In 10th class, you read the same topic. What I'm going to do. Now you have pyruvic acid. Pyruvic acid 2 molecule. This pyruvic acid have 2-3 options. In case of bacteria, or in case of human muscle, when we have shortage of oxygen, then fermentation process will happen and lactic acid will form. Lactic acid also have 3 carbon. Fermentation happen. And in this case, whatever NADH you produce during the reaction, no use. कोई काम की चीज नहीं है. क्यों? इससे एटीपी नहीं बना सकते. माइटोकॉन्ड्रिया ही नहीं है इस प्रोसेस के लिए, या ऑक्सीजन ही अवेलेबल नहीं है फॉर ईटीएस, तो ये यूज ही नहीं हो सकता. इफ इट कांट बी यूज, what will happen? Your NADH will again convert into NAD. Understood? And because lactic acid is formed, during this lactic acid formation, hydrogen is removed from NADH. This process, this enzyme involved here is known as lactate dehydrogenase. Okay, so lactate dehydrogenase. What is lactate dehydrogenase? Formation of lactic acid in which hydrogen is also removed. That's why dehydrogenase. Any doubt, guys? Then, enough. Second possibility that you studied in 10th class, this another process of fermentation in case of yeast, again fermentation will happen, and in this fermentation process C2H5OH and CO2 is removed. Correct? So can you say from pyruvic acid CO2 is removed? From pyruvic acid CO2 is removed. That's why it is known as pyruvic acid or pyruvate decarboxylase. Why? Removal of CO2 from pyruvic acid. Removal of CO2 from the pyruvic acid, or देखिए बैठे को चल लगे, or alcohol is produced. That's why another name of the same enzyme is alcohol is produced by removal of same hydrogen, dehydrogenase. Correct? So how lactate dehydrogenase same alcohol dehydrogenase? Correct? And because from pyruvic acid CO2 is removed, that's why pyruvate decarboxylase. Hello? Any doubt, guys? Same process will happen in this case. Your NADH, what was produced in the glycolysis process, have no Hello. Hello. So if you can hear me guys.
यस सर आ रही है आवाज
हाँ सर
ठीक है, तो समझ में आ रहा है अच्छे से, कुछ मिस तो नहीं किया? देखो अरे ये क्या है? तो ये पता चला कि हाउ एंजाइम आर नेम्ड. एग्जाम्पल, if you see this, lactate is produced by the process of dehydrogenation, removal of hydrogen. Alcohol is produced by the process of the removal of hydrogen. Same, hydrogen is removed. Second, pyruvic acid will undergo decarboxylation, removal of CO2. This is the second option. Third option, third fate of the pyruvic acid is, in case of Krebs cycle, it will convert into 6CO2 plus H2O, correct? 6CO2 plus H2O plus energy. And we have to read that topic only. हमें यही टॉपिक पढ़ना है, Krebs cycle आगे, correct?
तो
Yes, Anvita.
ये लैक्टिक एसिड और इथेनॉल एक टाइम पे वो हो सकते हैं, साइमल्टेनियसली फॉर्म हो सकते हैं, या फिर मतलब ऑल्टरनेटिवली फॉर्म होते हैं, मतलब जब जिसकी नीड होती
Depend करता है. Example, in case of yeast, this will produce. In lactic acid bacteria, lactate will produce. In case of muscle, if we have shortage of oxygen, then lactate will be produced.
है। डिपेंड्स ऑन द ऑर्गेनिज्म, मतलब किसको किस
Yes,
चीज
depend upon the condition.
का।
Correct. Second, if you study in this case, very less energy is produced. How much? Less than 7% energy is produced in the whole process. Correct. And you have hazardous chemical in this case. Example, lactic acid will cause pain in the muscle. Alcohol will make you, rosy. But I don't know what is the right name. ये अल्कोहल पीने से क्या होता है? You know that. Hazardous chemical. Next, in case of yeast, if you have 13% alcohol, then yeast will die. So you cannot produce natural alcohol that have more than 13%, because otherwise yeast will die, no more production, right? So these are the few number which is really, really important. Question, how much energy is produced? Only 2 ATP because of the glycolysis. Only this energy produced because of glycolysis, nothing else. Any doubt? So tell me guys, how is it going? Aditya, समझ में आ रहा अच्छे से?
जी सर, बढ़िया समझ आ रहा।
आदित्य बहुत लो है वॉइस.
सर
हाँ
बढ़िया
मनन,
आ रहा है, सही समझ आ रहा।
सही समझ में आ रहा है. अब मनन,
सर आ रहा है समझ में।
अन्विता,
यस सर।
समझ में आ रहा ना अच्छे से?
हाँ यस, समझ में आ रहा है।
प्रतिज्ञा, प्रतिज्ञा,
हाँ सर।
देवांश,
यस सर आ रहा है।
जॉर्डन,
Yes, understood now.
निर्जला,
यस सर, अच्छे से आ रहे हैं समझ में।
साहिल शर्मा,
सर जी आर पार
ओके. तो आगे आर पार नेक्स्ट टॉपिक करते हैं. तो नाउ वी हैव पायरुविक एसिड प्रोड्यूस इन साइटोप्लाज्म, है ना, by the glycolysis process. In case of aerobic respiration, because remaining whole thing we will study related to only aerobic respiration. Okay, anaerobic respiration is done. Only for the aerobic respiration we'll study. Okay, so what will happen in the aerobic respiration? You have pyruvic acid in cytoplasm. This pyruvic acid, when enter into the mitochondria, what will happen? It will try to produce mostly CO2. Okay, this happen in the Krebs cycle. Okay, and in mitochondria, same pyruvic acid try to produce ATP. This will be ETS, electron transport system. Okay. So first it will enter into the mitochondria, that is for sure. From cytoplasm, it will go to the mitochondria. Next important step is link reaction. Link reaction.
तो लिंक रिएक्शन कहाँ होता है?
माइटोकॉन्ड्रिया में.
माइटोकॉन्ड्रिया में ही ना?
हाँ. So as we already said कि pyruvic acid from cytoplasm is already transported. So you have two molecule of pyruvic acid in mitochondria. Coenzyme A1 chemical will be added. It will react with NAD positive. Okay. And in this process, what will happen? Magnesium ion is really important to process. And pyruvate dehydrogenase will be used. From the pyruvate, removal of hydrogen, dehydrogenase. Removal of hydrogen from the pyruvic acid. Removal of hydrogen from the pyruvic acid. क्यों? Because you know हमारे पास है NAD पॉजिटिव, ये बन जाएगा NADH, तो इसमें हाइड्रोजन आए कहां से? पायरुविक एसिड ने रिमूव किए. How we got hydrogen in NADH? From the pyruvic acid. That's why enzyme name is pyruvate dehydrogenase in presence of magnesium ion. So what will be the product? 2 pyruvic acid will convert into 2 acetyl coenzyme A, 2 carbon molecule. So what will happen to 1 carbon? It will produce CO2. What will happen to NAD? It will convert in NADH and H positive. 2 molecule. Okay, 2 molecule. Any doubt?
No, sir.
तो
And this is
ATP
link reaction. Huh?
तो सॉरी, टोटल एटीपी काउंट में ये वाला NADH का भी काउंट होता है ना?
बिल्कुल डेफिनेटली होता है, तभी तो हम इसको यूज कर रहे हैं, स्टडी कर रहे हैं.
बट फर्मेंटेशन वाला पार्ट जो है उसका कुछ रिलेशन नहीं है ना?
नहीं, फर्मेंटेशन वाला तो वो तो डिफरेंट ऑर्गेनिज्म में है ना,
हाँ
इसलिए.
ठीक. नहीं, मैं उसी में कुछ कंफ्यूज हो रही थी जब मैं पढ़ रही थी.
हाँ बिल्कुल, तो ऐसे में कई बार वो होता है ना कि अच्छा उसको क्यों नहीं काउंट करें, बिकॉज वी आर स्टडिंग डिफरेंट ऑर्गेनिज्म. फर्मेंटेशन ईस्ट में बैक्टीरिया में हो रही है, तो
हाँ.
that is the different process.
यस सर.
And this process, because there is a removal of oxygen and there is a oxidation of pyruvic acid by removal of hydrogen, that's why this step is also known as oxidative decarboxylation. You are removing CO2. You are removing CO2. And while you are removing CO2, it will undergo oxidation. Correct? While you are removing CO2, it will undergo oxidation. It will undergo oxidation. Correct? How do we know? Because removal of hydrogen is oxidation. So there is an oxidation process by removal of CO2 as well, because CO2 is also removed. That's why oxidative decarboxylation. So out of 4, 2 steps are done. Okay, out of 4, 2 step done, 1 glycolysis done, second link reaction is done. I will write this name. So this second step, link reaction done. First step was glycolysis. First step was glycolysis. Second step was link reaction. Third step will be Krebs cycle. Oxidative decarboxylation and glycolysis. Come to this topic. So, in case of Krebs cycle, because it was given by the scientist Krebs, first product is citric acid. That's why first product is citric acid. That's why this cycle name is also known as citric acid cycle. This citric acid contains 3 carboxylic acid. That's why another name of this cycle is tricarboxylic acid cycle, because citric acid contains 3 COOH. Correct? Contain 3 molecule of COOH, carboxylic acid. Correct? So first product is this. And first member, if somebody asks what is the first member in this process, member is oxaloacetic acid, which is 4 carbon molecule. Okay. How? Example. We had 2 molecule of pyruvic acid react with coenzyme A plus 2 molecule of coenzyme A plus 2 NADH, NAD positive.
सर थोड़ा सा
And
ऊपर Thank you.
so in this case, your pyruvic acid convert into your 2 molecule of acetyl coenzyme A. Acetyl coenzyme A.
सर ये लिंक
Along,
रिएक्शन पार्ट है ना?
हाँ, ये लिंक रिएक्शन को ही दोबारा लिख रहे हैं हम.
हाँ.
Along with that, 2 NADH is produced because 2 NAD were consumed. Na? 2 NADH produced, 2 acetyl coenzyme A produced. Now we will do only one cycle of acetyl coenzyme A. It means for 2 acetyl coenzyme cycle will repeat 2 times. Okay. So what happen? First, it will react with oxaloacetic acid, first member of this cycle. Both together in presence of water, by removing carbonyl coenzyme A, and because citric acid will produce, so citrate synthase is the enzyme used here. Citrate synthesis enzyme used here. What will be the product? Product will be citrate or citric acid, same meaning. So first product is citrate, 6 carbon. How 6 carbon? 2 carbon in acetyl coenzyme and 4 carbon in oxaloacetic acid. Total 6. Correct? This citrate convert into the isomer, that is isocitrate. Isocitrate, 6 carbon again. This isocitrate again convert into alpha ketoglutaric acid. I will make it big. alpha ketoglutaric acid.
सब by decarboxylation ना?
हाँ, alpha ketoglutaric acid. So in alpha ketoglutaric acid, you have 5 carbon. Logically, when 6 carbon present in one compound and another compound is 5 carbon, it means you definitely removed one CO2. And whenever you remove CO2, what will happen? Your NADH will also produced. Very, very, very important. Wherever you remove CO2 and NADH. This is decarboxylation process. Why? Removal of CO2. Removal of CO2, that's why decarboxylation. Also, this is oxidation process. Why? Because isocitrate will produce 2 hydrogen. These 2 hydrogen will be added in NADH. So this is oxidation process. Okay. This acetyl alpha ketoglutaric acid again remove one more CO2 and again produce one more NADH and H positive. Correct? So Jordan, tell me, what should be the number of carbon in the next compound?
It should be 4, sir.
Devans, if this have 4 number of carbon, is this oxidation process or reduction process?
Oxidation.
Yes, oxidation. 4 carbon, oxidation. Why? Because same like isocitrate produce alpha ketoglutaric acid, hydrogen is removed from isocitrate. How do we know hydrogen is removed? Because 2 hydrogen is added here. Somebody removed, that's why it is added here. Same here. If NADH produced and hydrogen is added, it means another molecule removed NAD. And sorry, remove hydrogen. Removal of hydrogen is oxidation. Now, this succinyl coenzyme A will convert into succinate. And when this thing happen, your GDP will convert into GTP. GTP convert into GTP. Later on, what will happen? It will produce ATP in the simplest term. Because ATP is produced in this process, yes, Tanvi, I was taking class of respiration. What happened? Tell me. So guys, ATP is produced. This step is known as substrate level phosphorylation. It means phosphorylation means formation of ATP. Phosphorylation means formation of ATP, and substrate level phosphorylation means formation of ATP without light. Some substrate form the ATP. Not, it is not produced in the final step, not in the initial step. In between, intermediate ATP produced. Substrate produce the ATP. Substrate level phosphorylation. Example, in photosynthesis chapter, we had photophosphorylation, formation of ATP in presence of light. This is not in presence of light, so substrate produce ATP. That's why substrate level phosphorylation. Even if you see here in the glycolysis, you produce ATP. That is also substrate level phosphorylation. This is also substrate level phosphorylation. Where is ATP produced? Substrate level phosphorylation. What does it mean? ATP is produced from the substrate. ATP is produced from the substrate. Understood? Succinate, because 4 carbons, so no CO2 removed. Correct? No CO2 removed in this case. Now, this succinate will convert into fumarate, though this word is not given in NCERT. But in this process, very popular enzyme and very popular molecule is produced, FADH, like NADH. FADH is produced. Correct? FADH is produced in this case. And in this process of FADH formation, what will you see that one popular enzyme is involved in this case, known as succinic dehydrogenase, complex 2 of electron transport system. And same question was asked in NEET 2025, another name of complex 2, succinic dehydrogenase. Correct? Because FADH is utilized in this case. Correct? FADH is utilized in this case. And one more thing, because again hydrogen is added in FADH, this is also oxidation. Wherever NADH is produced, oxidation. Where FADH is produced, oxidation. So 3 oxidation you can see here as of now. Okay. Fumarate convert into malic acid. Malic acid, 4 carbon again, same. Fumarate also 4 carbon. And ultimately, this malic acid convert into oxalic acid with the help of NADH.
सर मैलिक एसिड बनाने के लिए FADH2 से FAD प्लस बनेगा ना?
मैलिक नहीं, फ्यूमरेट बनाने के लिए ये बने।
अच्छा, तो मतलब FADH प्लस से FAD बनेगा फ्यूमरेट बनाने के लिए?
हाँ, FADH वहां पे प्रोड्यूस होगा तो
ठीक है।
फ्यूमरेट बनाएंगे। हाँ। And in last process, NADH is produced in presence of H positive. Correct? So again, this is oxidation process. Again, this is oxidation process. Why this is oxidation process? Because NADH is produced. So whenever NADH or FADH produced, it is oxidation process. You will ask कि सर हमने पढ़ लिया, समझ में भी आ गया, पढ़ा क्यों? Why do we study this topic? What is the purpose? Answer is because benefit of this process is you removed all this CO2. What is one of the product in your, what is the ultimate reaction in case of respiration? Removal of CO2. Correct? So we remove CO2 with the help of Krebs cycle. Understood? So in Krebs cycle, we removed all the CO2. What is the benefit of this reaction? Because you use 2-pyruvic acid. From 2-pyruvic acid, you produce total 8 NADH because 2 cycle. 1 cycle, 4 NADH. 2 cycle, 8 NADH. 1 cycle, 1 FADH. 2 cycle, 2 FADH. And 2 cycle, 2 ATP. This is the conclusion of the reaction. Understood?
फिर एक बार ऊपर
NCERT mentioned कि we did so much from this chapter, still not many ATP produced as of now, right? Lot of energy is not produced. Oxygen is not coming into the picture because we know one of the important reactant of the process of respiration is oxygen, O2. But oxygen is not coming into picture as of now. Correct? Everything is without oxygen. How? And what is the use of oxygen if it is not coming into picture? Last important thing of this chapter will do electron transport system. So tell me guys, raise your hand key. You understood everything what I taught as of now? Aditya, yes. Nirjala, yes. Other people?
Yes, sir. I'm not able to raise my hand though.
Okay. Jordan, 3, yes. Anvita,
हाँ सर
4. Pratigya, Manan, Devans,
Yes, sir.
Komal, and Sahil Sharma.
What, sir?
समझ में आया अच्छे से?
हाँ सर।
हम्म
आ गया सर।
तो अब तक जो हमने ये पढ़ा था समरी वो हमारी यूज हो गई है. एज ऑफ नाउ वी डिड ग्लाइकोलिसिस. One glucose molecule produced 2 ATP net, 2 pyruvic acid and 2 NADH. In link reaction, 2 pyruvic acid used and 2 acetyl coenzyme A, 2 NADH and 2 CO2 produced. In Krebs cycle, also known as TCA cycle or citric acid cycle, when these 2 acetyl coenzyme are produced, so it will release all the CO2. It means 100% CO2 is removed. How? Because 4 CO2 removed here and 2 CO2 removed here, total 6. 1 glucose produce 6 CO2, done. 6 NADH in the Krebs cycle, 2 FADH and 2 ATP. Now, no more ATP, no more reaction for CO2, no reaction for anything. You have conclusion of, as of now, if you see, ki what do you have? You have nothing except total 10 NADH available from 1 glucose, 2 FADH from 1 glucose, and 4 ATP from 1 glucose. Correct? Or these things produced from the 1 glucose. Correct? 1 glucose produce so much thing. All other are utilized. CO2 are removed. Correct? Because we know 1 NADH produce 3 ATP, so 10 NADH produce 30 ATP. 1 FADH produce 2 ATP, so 2 FADH produce 4. And 4 ATP as such. Total number of ATP produced in the whole process, 28 ATP. How these 38 ATP will be produced? Explained by the last and most popular reaction, Krebs cycle. Correct?
सर
Like, yes,
सर 2 मिनट के लिए स्क्रॉल डाउन करिएगा.
बिल्कुल. Electron transport system, ATS. And whenever I take counseling from anyone, I ask question from this topic only. ETS. बिल्कुल निर्जला.
ओके सर.
ETS. कितने लोगों को, how many of you had so much fear from this electron transport system or in general this chapter respiration in plant की? ये तो बहुत टफ है, मतलब देखते ही एकदम. Jordan, you still have?
Not now, but then like I had it before.
How many of you guys still have this issue?
अब नहीं है सर, पहले था.
ठीक है, अब नहीं है पहली बार. तो produce 38 ATP produced here ultimately. Oxygen consumed ultimately in this step. H2O is produced. Other people, Anvita, do you have fear from this chapter?
The 11th में था, because you know who taught it, but not anymore.
Okay. So this is the conclusion of the process. Because this is a complex process, we divide into 5. And to explain in easy way, I call this electron transport system is the bank process. How you go to the bank and multiple thing. How it is managed, like you go to the bank and manage the thing, correct?
सर आप वो डीडी वाले एग्जाम्पल से समझाएंगे क्या, डिमांड ड्राफ्ट वाले?
हाँ
सर एक बार नॉर्मल से भी समझा देना.
बिल्कुल, दोनों से समझाऊंगा जिससे इजी रहे. सो एग्जाम्पल गाइस कि NADH is your check. When you deposit check in your bank, you get 3 lakh rupees. Here you get 3 ATP. Okay. If you deposit your DD in your bank, FADH, then you get 2 lakh rupees. And if you have cash, no need to deposit cash because cash is cash. Ultimately, you need cash. So your ATP is your cash. Okay. And when you deposit your demand draft or check, ultimately you want to get cash from the bank. Okay. Understood. There are 5 complex in the step. Complex 1, where complex 1 is like counter 1. You go to the bank, go to counter 1, and on counter 1 you deposit your check, NADH. And to process this NADH, there is an enzyme, NADH dehydrogenase. Correct? But if you have DD, you go to counter number 2 and you deposit your DD. DD is FADH. So this is known as FADH dehydrogenase. You can call it as, but NCRT mentions succinic dehydrogenase. Same thing. Because in the process of the FADH, succinic dehydrogenase enzyme is used. Remember this FADH. मैं भी चाय पीऊंगा. Okay. Okay. Any doubt, guys? Still here?
आपने FADH क्यों लिखा?
क्योंकि अल्टीमेटली इसमें FADH ही यूज होता है. जैसे इसमें NADH यूज होता है, इसमें FADH यूज करता है. Okay. Then you go to count, like now it's not your job now. It's a bank process. If like on counter number 3, both your check and DD will be checked properly. If everything is okay, like can we proceed further? So for this you have one popular cytochrome BC1. Okay, this is counter number 3, where you can process both DD and check. Then it will go to the manager room, counter number 4. Complex 4 is counter number 4, where in the manager room, this is manager room, it will be checked properly and signed by the manager. Only then you can get it. And the name of this is cytochrome C oxidase. Cytochrome C oxidase. Cytochrome C oxidase is known because in this whole process, one person is very crucial, manager. And this manager is cytochrome C. which is responsible for everything. That's why for manager you have special name, cytochrome C oxidase. Ultimately, it will go to the cash counter complex 5. Cash counter. Where ATP is produced, you will get cash. And this is known as ATP synthase. Synthesis of ATP on your cash. Like, because now it got signed by the manager, it will go to the cash counter. From cash counter, you will get cash. It means you will get ATP. Tell me, guys, everybody, does it look very easy or is it confusing? बोल दो सर मजा आ गया. Too good.
Good.
Best. Explanation ever. बस थोड़ा सा मोटिवेट कर दो मुझे कि सर वाओ.
हाँ सर मजा आ गया.
Everybody.
It was
अरे
good.
Adit, it was good. Simple and sober.
Sure.
So raise your hand if you understood every single thing, because only then I'll process to the whole thing. Anvita, Nirjala, Aditya, Bubbly, Komal, Manan,
Yes, sir.
Devans,
Yes, sir.
Jordan, and Sahil Sharma.
Yes,
Is it really difficult anymore?
not much.
Because most of the question asked from this part only, na? Ki what is the name of every enzyme? NEET 2025 question was succinate dehydrogenase. What is the another name of complex 2? That was the question. And in this case, you know, complex 2 is succinate dehydrogenase. As simple as that. Sometimes it asks what is the counter number 3? BC1. Counter number 4, cytochrome C oxidase, because cytochrome C is involved, main person. And from the cash counter, you will get cash. Cash is ATP. Yes or no, guys? Give me 1 minute. I'll be back and I'll finish this part. So, first question was this: complex 2 of mitochondrial matrix succinate dehydrogenase. अब ये देखो, from biomolecule there were 5 questions in NEET 2025. Biotechnology there were 6 questions from in NEET 25. From respiration in plant there was 1 question in NEET 2023. Right? Only 1 question. This is see this question. Anvita, citric acid cycle.
Sir, mitochondrial matrix.
Yes. Devans, glycolysis. Devans.
Yes, sir.
All of you write your sequence. All of you write your sequence in chat box. A is this, B is this, C is this, D is this. अच्छा एक important point था मैं notes में add कर दूं. I'll add in notes. In electron transport system, this system occur in where is pen? So this electron transport system occur in inner mitochondrial membrane. Very, very important. Inner mitochondrial membrane. Because we should know where every reaction happen. Inner mitochondrial membrane. Correct? So हाँ. देवांश, are you there? मेरी आवाज आ रही है? Jordan, glycolysis.
Cytoplasm.
Okay. Nidula, electron transport system.
Sir, inner membrane space of mitochondria.
Yes, and proton gradient.
Inner mitochondria.
Inner mitochondrial membrane. How many people are there? Aditya, Anvita, Pratigya, Manan, Devans, Jordan, Nirjala, and Komal. Okay. Komal, this question. Step in TCA cycle that do not involve oxidation. Komal. Answer will be, all of you have to write, I'll check after this. Because I mentioned this in the class, I taught where oxidation happen. Wherever NADH or FADH produce, oxidation happen. NADH or FADH produce, oxidation happen. So what will be the answer? C. Succinyl coenzyme A2-succinic acid, ATP is produced. No NADH, no FADH produced in this case. Very popular question. Very important question. Popular why? Because give you new perspective. Okay. So answer will be C. And I'll check after this because Aditya, ATP is used in the two-step in the glycolysis. Is this correct? Aditya. Anvita, ATP is used at two-step in the glycolysis is correct. And first ATP, glucose to glucose 6-phosphate. Second ATP, fructose 6-phosphate to fructose 1,6-phosphate. Both correct and correct. Explanation. Why use in two-step? Because this is the explanation. Correct. So correct explanation. Any doubt? So we can come to the electron transport system. So you can add this point here also. K ETS occur in inner mitochondrial membrane. Inner mitochondrial membrane. Okay, inner mitochondrial membrane. Move to the what happened in the ETS. Diagram is important. Example. This is the bank. In case of bank, the first and this bank occur in inner mitochondrial membrane. This is proper inner mitochondrial membrane. This is matrix, and this is intermembrane space. Inter means between. Between two membrane, you have space, intermembrane space. Okay. So what will happen in this case? If you have NADH that consume in complex 1, correct? So what will happen? This is complex 1. NADH dehydrogenase is used. FMN will enter. Go to the common carrier FES. याद रखना everybody, this is very popular. Every time you will have iron. Why you have iron? Because in case of iron, it can easily convert into +2 and +3 oxidation state. तो जो जिसके अंदर भी variable oxidation state होती है, wherever you have variable oxidation state, this is really good carrier because it can transform from one form to the another form. Okay, what happened? 4 people wrote. Nirjala is correct, both correct. Okay. So, in this case, what will happen? It will produce hydrogen further because hydrogen is responsible for the, ATP formation. Passage of hydrogen is responsible for the ATP formation. From FES, it will go to ubiquinone. If it take hydrogen, it will convert into ubiquinone. Addition of hydrogen is quinol. This is quinone, and it means it is ketone. Addition of hydrogen means reduction. Reduction of ketone will produce alcohol. Ubiquinol. Correct?
So what is FML?
Flavin mononucleotide.
Okay, sir.
Then after this, you have cytochrome B. Cytochrome B. Then again same FeS, iron sulfide. Again C1. Okay. So that's why this complex is known as cytochrome BC1. Correct? This is complex 3. Cytochrome BC1. Cytochrome BC1. Correct? So from here hydrogen will go like this. If complex 2 is given, it is succinic dehydrogenase. Succinic dehydrogenase. In succinic dehydrogenase, FADH is consumed. What will happen? First, receiver, her count, like wherever you go to the counter, every counter will have different person. That's why first person on the first counter is FMN. First person on the third counter is cytochrome B. First person for the FAD will be different. It will be FAD. Divinide adenine dinucleotide, FAD. Further process is more or less same. It will go to FES. From FES, it will go to ubiquinone, convert into ubiquinol, and from this hydrogen will be go there. Correct? Hydrogen will go there, and electron can pass through this. Electron can move to this. Electron can move to. After this first point is the things happening in case of counter number 1. Okay, counter number 1, you deposit your check, person will receive, and with the help of helper, like there is one common guy who transport the check, DD, everything, all the document, helper to the manager room. Correct? So helper is common, FPS. Helper is common, FPS. Helper is common, FPS. Correct? Same thing happen if you give FAD, common helper, FPS. Third counter, common helper, FPS. This common helper will go to the manager, and main manager is cytochrome C. Cytochrome C. This cytochrome C will present on the outer side of the membrane, outer surface of the membrane, and then it will go to the next complex, complex number 4. And because manager is more important here, that's why in counter number 4, it is also known as cytochrome C oxidase. Manager will be influenced. So in this case, you will have copper A, cytochrome A, cytochrome A3, and copper B. Copper A or copper B. Okay. Copper A3. These are the people sitting in the manager room taking care of that. If DD or check come to their room, they will cross check it. इनका काम है क्रॉस चेक करना कि जो डीडी लोगों ने चेक किया है, इज इट एवरीथिंग फाइन. Correct? And the most popular event happen here कि when oxygen react in this process, water will be produced. Very, very, very important, sir. And NCRT mentioned कि as of now, no role of oxygen. Limited role of oxygen was not limited. No role of oxygen was there as of now. So oxygen is used in the last step, terminal stage. Last step in complex 4, oxygen is used and H2O is produced. Correct? And ultimately, wherever you are producing hydrogen, these hydrogen will be used in the last step, complex 5. F naught, F1, F1, F naught. Correct? So when you have lot of hydrogen here, negative charge on the another side. Okay. So when 4 hydrogen pass through this, correct? Here you have complex 5, counter number 5, cash. Correct? Cash. So cashier, complex 5. This name is ATP synthase. Synthesis of ATP. And when ATP synthesis occur, ATP will produce. Done. This was the bank process. Any doubt, guys?
No, sir.
But remember, all these things you will be like you will get better if you already are aware about this and you just need quick revision. Correct. And this important topic is done. But imagine people who feel कि this chapter is really difficult, you get only 1 or 2 questions. बस यही इतने ही आते हैं, या तो 1 question या 2 question. Any doubt, guys? So what is happening ultimately when NADH or FADH are used, consumed in a reaction, NADH or FAD are consumed in reaction. This is responsible for formation of ATP. This is the formation responsible for formation of ATP. And ATP formation because of the oxidation is known as oxidative phosphorylation. So, in case of oxidative phosphorylation means process of formation of ATP. Process of formation of ATP due to the oxidation. Because of oxidation is oxidative phosphorylation. How oxidation? Because when your NADH plus H positive convert into NAD, what is this? Removal of hydrogen. What is the removal of hydrogen? Oxidation. Correct. So because of oxidation of NADH, ATP will form. Same way, FADH. FADH when react with H positive ion, oxidation happen. Same FAD positive produce same oxidation process. Correct. In chemoosmotic hypothesis, you have seen that this is in inner mitochondrial membrane. You have F naught. This is ATP synthase enzyme, which is F naught and F1. So when hydrogen pass through this, ATP will form. ATP will form. Correct. This is the last step, and your 90% job from this chapter is done, guys.
Sir,
Word oxidative phosphorylation is very, 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 very popular. And if you feel कि sir, what is this?
sir,
Phosphorylation means of ATP. Yes, Pratigya.
सर मेरा बहुत ज्यादा नेटवर्क इश्यू हो रहा है.
कोई बात नहीं, क्लास भी खत्म हो रही है बस 2 मिनट में. तो ऑक्सीडेटिव फॉस्फोरिलेशन में क्या होता है? ATP formation because of oxidation, as simple as that. So ETS, another name is oxidative phosphorylation, because here ATP is produced because of oxidation. Correct? For ETS you use word oxidative phosphorylation. Done. Okay. Sometimes they will not give you ETS, they will write oxidative phosphorylation. It means ETS only. Right, guys? Any doubt? Look
No,
at
sir.
this. Oxidative decarboxylation, not phosphorylation, decarboxylation. Okay. Oxidative decarboxylation, what is this? Link reaction. In link reaction, which enzyme is used? This enzyme is used. Write down your answer. Example, Glycolysis. Which enzyme is which? What is the another name of glycolysis? EMP pathway. Write down your answer, guys, in that box. Oxidative phosphorylation. It means ETS only. Now you got see 2023 question. Oxidative phosphorylation means what? ETS. Another name of this ETS. Tricarboxylic acid cycle, Krebs cycle. Which enzyme is used in Krebs cycle? Citrate synthase. Any doubt?
No, sir.
Done. This is the level of question. Do you feel
सर कोई
that
डाउट नहीं है.
this chapter look difficult, but questions are easy? Correct. Questions are very easy. How many times decarboxylation occur in case of TCA cycle? 2 times CO2 is removed in one TCA cycle. 2 CO2 are removed. Correct. 2 CO2 are removed. 2 CO2 are removed. That we'll discuss later.
सर मुझे एक चीज
5.
पूछनी है.
हाँ पूछो.
मतलब जैसे एक जो टीसीए साइकिल चलती है उसमें दो वो होता है, मतलब क्योंकि पायरुवेट जो होते हैं वो दो बनते हैं,
हाँ.
तो उस हिसाब से 4 डी कार्बोक्सिलेशन नहीं होना चाहिए.
लेकिन एक साइकिल की बात है ना. 2 पायरुविक एसिड में तो 2 साइकिल होंगी.
ओके सर.
हाँ. 5 carbon compound in TCA cycle, alpha ketoglucaric acid. Correct. You see question level. Number of decarboxylation of isocitrate in whole cycle 2. Isocitrate 1. क्योंकि आइसोसिट्रेट का तो एक ही बार होगा ना. आइसोसिट्रेट अंडरगो डिकार्बोक्सिलेशन 1 टाइम.
Yes.
अल्फा कीटोग्लूटारिक एसिड 1 टाइम. टोटल 2 टाइम.
बस वैसे 2 बार है ना टोटल.
क्या?
वैसे टोटल डीकार्बोक्सिलेशन 2
टोटल 2.
टाइम्स है ना.
आइसोसिट्रेट 1, अल्फा कीटोग्लूटारिक ग्लूटारिक एसिड 1.
हाँ.
Net gain of ATP when glucose convert into pyruvic acid
2.
2. Amount of energy released from glucose in lactic acid fermentation.
Less than 7%.
Exactly. Following incorrect. In the aerobic respiration, role of oxygen limited to the terminal stage. Correct.
True, sir.
EPS1 NADS produce 2 ATP.
Wrong. उल्टा लिखा है.
Exactly. You got answer. ATP synthesized in complex 5. Correct. Oxidation reduction produce proton gradient in the respiration. Correct. Proton gradient.
आपका से क्वेश्चन तो इजी है बट नंबर ऑफ क्वेश्चंस भी तो कम है.
हाँ, number of questions कम है. देख. Number of substrate level phosphorylation in one citric acid cycle or TCA cycle. Only one ATP is produced na in one cycle. From succinyl coenzyme A to succinate, right? GTP or ATP. Correct. Glucose convert into glucose 6 phosphate. First irreversible process catalyzed which hormone? Hexokinase.
हेक्सोकाइनेज होगा सर.
Correct. Where ETS located? Inner membrane.
इनर मेम्ब्रेन की जो माइटोकॉन्ड्रियल वाली होती है मतलब.
So class is done. Tell me, guys. Turn on your video. Let me stop this video.